Enzyme inhibition is the common mode of drug action. Non-specific inhibition. Many drugs are capable of denaturing proteins. These drugs alter the tertiary structure of an enzyme and thus inhibit its action. For example, heavy metal salts, alcohol, formaldehyde and phenol inhibit enzymes non-specifically. Specific inhibition. Many drugs inhibit a particular enzyme without affecting the others. Specific inhibition may be of two types. Competitive. The drug competes with a normal substrate for their attachment on the active site of the enzyme. Non-competitive. Some drugs do not bind to the active site of the enzyme, but bind to a different site of the enzyme, which is called allosteric site. This binding of the inhibitor at allosteric site alters the shape of the active site in such a way that the substrate cannot recognize it, and therefore enzyme loses its catalytic property. Receptors as drug targets. Receptors are proteins that recognize and respond to the body's own chemical messengers, such as hormones and neurotransmitters. Receptor proteins are located either on the surface of the cell or inside the effector cell. In the body, message between two neurons and that between neurons to muscles is communicated through chemical messengers, which are received at the binding sites of the receptor. To accommodate a messenger, the shape of the receptor site changes. This brings about the transfer of message to the cell. Thus, receptors perform two essential functions: recognition of messenger molecule, transduction of the signal into a response. There are a large number of different receptors in the body that interact with different chemical messengers. These receptors show selectivity for one chemical messenger over the other. An agonist is a drug which produces a stimulation-type response. It is a very close mimic of the natural messenger and fits with the receptor site and thus is able to initiate a response. An antagonist drug interacts with the receptor site and blocks the normal response for that receptor. Therapeutic action of different classes of drugs. Now we'll discuss the therapeutic action of few important classes of drugs. Antacids. Acidic pH is necessary in the stomach for digestion of food and good health. But excessive acidity in the stomach causes discomfort, heartburn, and gastric ulcers. The chemical substances which neutralize excess of acid in gastric juices and raise the pH in the stomach to an appropriate level are called antacids. For example, sodium bicarbonate and the mixture of aluminium and magnesium hydroxide are commonly used antacids. Antihistamines. Histamine is a toxic amine and is found along with proteins in nearly all tissues of the body. A number of sensitizing substances called antigens derived from food products, pollens, dust, human hair and sheep wool may cause the release of free histamine in the body, which causes symptoms associated with allergies, hay fever and the common cold. An antihistamine relieves these symptoms by blocking the action of histamine. For example, chlorpheniramine, brompheniramine and Seldane. Antihistamines interfere with the natural action of histamine by competing with histamine for binding sites of receptor where histamine exerts its effect. Neurologically active drugs. These drugs affect the message transfer mechanism from nerve to the receptor. Tranquilizers. These are the drugs used for the treatment of stress and mental diseases. These drugs relieve anxiety, stress, irritability and excitement. For example, chlordiazepoxide and meprobamate are mild tranquilizers. Derivatives of barbituric acid constitute an important class of tranquilizers known as barbiturates. They induce a state resembling to sleep. For example, Veronal and Luminal. Antidepressants. Noradrenaline is an important neurotransmitter that plays a role in mood changes. Antidepressant drugs inhibit the enzymes which catalyze the degradation of noradrenaline. For example, iproniazid and phenelzine. Equinal